Nucleotides are subunits of nucleic acids. Nucleic acids exist as polymers or polynucleotides. These also consist of monomers called nucleotides. Nucleotides are composed of nitrogenous bases, pentose, and phosphate groups. Nucleotides serve as building blocks for the construction of nucleic acids. Nucleotides composed of ribose are called ribonucleotides, and nucleotides containing deoxyribose are called deoxyribonucleotides. Bases are named after the fact that they are basic, not acidic, and they each contain several nitrogen atoms. Nucleotides can pair up with each other. Cytosine always pairs with guanine, and adenine pairs with thymine in DNA or uracil in RNA. The next major component of a nucleotide is the sugar. There are many kinds of sugar, but two are more important here. Ribose is the sugar you'll see in RNA. There is a version of ribose that has an oxygen atom missing, and we'll call that sugar deoxyribose. That's kind of a sugar in the DNA's nucleotides. Remember that DNA stands for deoxyribonucleic acid. The last major piece of a nucleotide is a phosphate. A phosphate is a phosphorus atom bonded to 4 oxygen atoms. The bonds between phosphates are very high energy and act as a form of energy storage. When the bond is broken, the resulting energy can be used to do work. Adjacent nucleotides are joined by covalent bonds called phosphodiester bonds. This results in a backbone with a repeating pattern of sugar-phosphate units. Phosphodiester bonds occur between OH group on the 3rd carbon end of the nucleotide and a phosphate on the 5 carbon end of the nucleotide. 2 free ends of the polymer are distinctly different from each other in chemical polarity. One end, the phosphate, is attached to the 5 carbon end, and the other end, the OH group, is attached to the 3 carbon end. RNA molecules are called single polynucleotide chains. The DNA molecules are called 2 polynucleotide chains that spiral around an imaginary axis, forming a double helix. Purine always binds with a pyrimidine in the helix through hydrogen bonds. Adenine forms 2 hydrogen bonds with thymine. Guanine forms 3 hydrogen bonds with cytosine. Therefore, given the sequence of bases on the strand of the DNA double helix, the sequence of the complementary strand is fixed and easily determined.